Peter. Okay. So, let's pick up where we left off. So we started talking about these dihybrid crosses, and I drew yeah, I want to do this. Sure, let's do it. Let's do a dihybrid cross. Because they're so weird. And they're hard. They seem really complicated and scary, but they're actually fine. Okay, so somebody give me a trait that we want to talk about. Let's talk about pea plants, because why not? Because we're still on Mendo. And let's talk about peas. Give me some traits. Tall. Tall? Okay, so we're gonna have some tall plants. Tall and short. Okay. What's another trait that we can use for our dihybrid cross? Color of
The plant.
the
flower.
flower. Okay. So then we'll do flower color. Okay. Let's decide, is dad gonna be let's remember what these words mean. Homozygous dominant. What does that mean?
Two dominant. Two T's.
Alleles.
Alleles, thank you.
Yes. Okay. What does homozygous recessive mean? Two recessive alleles, two little t's. And then what's the other one that you could possibly be? Heterozygous, and that means big T, little t, right? Okay. So is dad going to be homozygous dominant, homozygous recessive, or heterozygous? What do we think? For height. Okay, homozygous dominant. Okay, is dad going to be have purple flowers or white flowers? Purple. Okay, let's make him heterozygous because that's gonna be more interesting. Okay. Okay, so that's dad. Now let's do mom. What should we have mom be? Should we have mom be short? Sure. Okay, we're gonna do little t's, and I'm gonna try to make this be really obvious that these are very different. Okay. What color flowers does mom have?
White.
Okay. And that means that she's gonna be, ooh, that's gonna be really hard. Okay, I'm gonna make my lowercase p's look like this so that we can track them better. Okay, that's weird, but we'll do it. Okay, so what's the combination? So let's draw our little thing over here. Okay, right, so this is a dihybrid cross. So what combination of alleles can dad give us? So we need to pull one from here and one from here, right?
Big T, big T.
Okay, I'm gonna, hold on, I'm gonna try and keep my color coding straight. Big T, big P. Okay,
Big T, little T.
big T, little p, because these two could go together. Okay, these two could go together. Big and big, and then we'll have these two go together. So we'll have, oh, this is what I was gonna do this. Right, that's all the combinations of the wheels for these two traits that dad could give us. Now, mom's gonna be easy because she's homozygous recessive in both traits that we care about, so we're all gonna do little t, little p. Doesn't matter how we sort them out, they're always gonna be the same. Okay. So now all we have to do is bring these down. Okay. And you guys could fill that rest of that table out, right? We're confident in how Punnett squares, we can do that with Punnett squares. Okay. What information does that give us? Probabilities
Of the phenotypes.
of the, yeah, so probabilities of the phenotypes of the offspring of those two parents. It's not a guarantee that we're gonna have of these, if these two parents have 16 kids, they're not gonna all be, you know, it's not exactly that, because each time a spermie and an egg meet each other, that's an independent thing, just like rolling the dice or flipping the coin.
And that's called rule of multiplication, right?
So so if I was gonna say, okay, so in our example, I rolled the dice, right? And I said, okay, I got a 6-sided dice, so the probability of me getting a 6 is 1 in 6. Probability of this dice being a 6 is 1 in 6. So probability of both of them being 6, then we would use multiplication rule, just to multiply those fractions.
And that's probability of two independent events occurring simultaneously is a product of their individual probability.
That's correct, yeah. Yep. Okay. Great. So that's a good refresher on how to play Punnett squares and what it tells us. Okay, so there we go. And we can do this for multiple traits. Usually, like the simple Punnett squares, we usually just use one trait. So there we go. All right. And then so if we did this, we could find out that we already said all those words. Okay, so here we go. Principle of segregation, and principles and laws and rules are stuff that you want to write down. Okay, so this kind of goes back to meiosis. If we have two alleles for a gene, they're going to separate during gamete formation, i.e. meiosis, right? And are rejoined at random from each parent during fertilization. And we saw this with the coyotes that we pretend to mate, right? So our basis of allele segregation comes during meiosis. Which stage of meiosis do we see sister chromatids being separated? Anaphase, which anaphase? Anaphase 2. Yep. What happens, what separates during anaphase 1? The homologous chromosomes. Yep. Okay. All right. So Mendel didn't know about chromosomes and meiosis and stuff because they haven't been invented yet. I mean, they haven't been described yet, right? Of course they existed, and that's why he was able to see these patterns, but we didn't know about that process at during that time. So, but definitely his observations fit with the molecular and cellular processes that we understand now. Okay, principle of segregation. Our alleles are separating during meiosis. All right. So here's what Mendel's found out, that this blending thing that the traits are born through the fluid. Remember that? And it's like, what does that mean? All right. So we didn't really see this blending of traits because we saw that each of these traits were distinct and discrete from each other. And we see, and when we took two heterozygous individuals, in that first generation, we would see that recessive trait disappear. And then it would come back up in the second generation. And so this is like saying, okay, no, because if it was blending, we'd all be homogeneous and we wouldn't see those traits pop back up. Okay, so Mendel knew about Chucky D, but Chucky D did not know about Mendel. And Mendel was like, he didn't know, like, it was just like Picasso, like after he was dead, and everybody was like, actually, that was like really cool, that guy did. And then of course, Mendel's work really helped support the theory of evolution. The theory meaning the scientific term of the theory, meaning lots of evidence for it, and not just something that you thought of on the throat this morning. Okay, there we go. All right, so when does Mendel's stuff not really work out? Oh, most of the time. So here's Mendel's assumptions or Mendel's model of inheritance is assuming there's three things. Oh man, list of 3 to 4 or 5 things. Damn, they sure really make great multiple choice questions. All right. So Mendel's genetics assumes that one trait is controlled by one gene. Each gene has only 2 alleles. Look around this room and look at everybody's hair color. There's more than 2 hair colors here, yeah? Okay, all right. And then there's a clear dominant and recessive relationship between these 2 alleles. But, you know, sometimes it doesn't work out as well, right? Something else is happening if we don't see the mathematical model that Mendel showed us. If there's something else going, like if they don't meet those ratios, something else is happening. So what else could there be? How else, what other kinds of inheritance models are there? Well, let's talk about those. We might have traits being carried on and being controlled by more than one gene. We might have one gene controlling more than one trait. Remember, we talked about that when we were talking about gene regulation. One gene can go on to produce multiple proteins. We do that by during RNA splicing and chunking out one of those exons. Hopefully you guys know what that word exon means. Right? We have often we have multiple alleles, more than 2 alleles. Hair color, for example. We talked about coat color in cattle last time. And we might have incomplete dominance and codominance. Ooh, what are those? So if we see, so codominance, if we see a heterozygous individual, they'll be somewhere in between. So, you know, I think the easiest example is to have a tall plant and a short plant. And if we see a medium-sized plant that's heterozygous, and that would be codominance, right? That would kind of sounds more like that blending of traits idea that we had before. Okay, so in order to get to the phenotype of whatever critter is that we're talking about, so that's our phenotype is the measurable traits, right? Or the physical characteristics of the thing is a combination of their genes, epigenetic controls, and environmental conditions. So just because we want it to be simple, we want there to be 2 choices for seed coat color or plant height or whatever. The real world is far more complicated than that. And so if we are interested in understanding why things express the traits they do, we need to understand and be able to control for these other factors. That's why cloning, using trees that clone each other themselves is really handy. That might be an essay question or something. Okay. Because that allows us to control for genotype and epigenetics, and then we can vary the environment. And then any changes that we see to the traits of those trees or whatever, then we know it's due to environmental conditions. Or if we take a bunch of trees from a whole bunch of different places that have a whole bunch of different genes, and we grow them in the same environment, now we're holding the environment steady and changing the genes, then we can say, okay, well now we know how important genes are. Okay. All right, polygenetic inheritance. And this is when we have multiple genes controlling the same trait. these traits seem to have a continuous variation. So the good example is height for humans. On average, women are 161 centimeters, and that's like what, 5'3" or 4" or something. Men on average tend to be a little bit taller. There's quite a bit of overlap, and you know, us people that are down here, yeah, we're the 1%. Yeah. So we actually see this on a continuous basis. So instead of having these discrete categories, blue or red or purple or white flowers or smooth and wrinkled seed coats, we see this gradation. And we tend to see this normal distribution, this nice little bell curve that most people are somewhere in here. A few people are really short, a few people are really tall, etc. Okay. Wait. All right. Pleiotropy. I always have to look this word up because I'm like, what does that mean again? This is where we have one allele that has more than one phenotypic effect. Right, so one gene is controlling more than one trait. And that's hard to predict, right? Because maybe we don't fully understand what each allele is controlling in our bodies. So cystic fibrosis, sickle cell anemia, where we can have multiple symptoms being traced back to one allele. We can have more than one allele for each gene in the population. So the good example of this that you guys are probably familiar with is blood type, where we have the A allele, the B allele, and the O allele. So each individual can only have 2 alleles, right? Because we're diploid organisms. But what you get, the pool that you can choose from is now larger than what we saw with peas. Okay. I think that's all I want to say about that. Okay, incomplete dominance. This is where we see the I messed this up. I always mess this up. Heterozygous is the intermediate of the phenotypes between the 2 homologous phenotypes, right? So we have a tall plant that's homozygous dominant. We have a short plant that's homozygous recessive, and then our heterozygote is somewhere in between. Or we see this. This is kind of that blending that we talked about. Red flowers crossed with white flowers make pink flowers. That's due to incomplete dominance. So if you guys mix these up, I will understand. So codominance shows that the heterozygous have some aspects of each phenotype. So the type AB blood, and so probably some of you are more familiar and more well versed in blood type than I am, but you know, the receptors on your red cells are determined by your blood type, and so people that have both alleles have receptors of the A type and receptors of the B type. Yep, you can look at this if you're interested in blood types. And then of course, as always. Sorry, Carlos, or Carson, I wasn't watching the screen enough to let you in, so hopefully. we're, hold on, let me wait till he connects to audio, so when I talk to him, he can hear me. Well, okay, so, of course, just like we saw in that little equation, we always have environmental influence on the way that our traits are expressed. So this really great example of, in certain breeds of rabbits and cats, their coloration is due in part to environmental factors. So the thing that the protein, the pigment that gives you dark coloration in all sorts of critters, from humans to fungi, animals, and plants, all the things, use melanin. And so in this breed of rabbits and this breed of cats, there is an enzyme that breaks down the thing that produces melanin, and it's active at certain temperatures. So the parts of the cat that are cooler can produce melanin. And so you get these little dark patches on these different, these cats and stuff. And that's due to the inactivity of an enzyme that is breaking down melanin. And so here where the cat is a lighter color is because that enzyme is breaking down the melanin. It's not being produced. And look, you can see on his little legs, you can see a little gradation. It's so cute. All right. Okay. All right, that's where I'm going to end this part of the blah blah today. How are you guys feeling? Do you guys want to go straight into the review, or do you guys want to take a little break, go get some coffee or whatever? We still got coffee. Okay, well, let me put Carson, I'm going to put you over here. Sorry that I didn't get you admitted immediately. I was watching and okay. 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 Let me put this back around.